All right, recording has started. So our next presenter is from the CNAS Emerging Scholars work. This is Gayatri Raut. Can vitamin B1 supplementation ameliorate non-alcoholic fatty liver in mice?
Hey, hi, I'm Gayatri. I'm a third year in cell molecular and developmental biology, and we're going to talk about vitamin B1 supplementation and if it'll ameliorate alcoholic fatty liver in mice. So to kind of understand our research a little bit, you have to know about the liver and soybean oil. So the liver is a place of fat storage. It'll break down fat and convert excess fat into carbohydrates and proteins that can be used to store later. The liver is also continuously producing bile, which is a substance that helps convert fat into energy. And the liver also gives us clues about diet-induced obesity and metabolic disease. And so soybean oil is relevant because it has started to become increasingly popular over the past couple decades. Currently, soybean oil is the most widely consumed edible oil in the United States, and its economical costs and neutral flavors make it a convenient choice for consumers and producers alike. And soybean oil has also been linked to the prevalent obesity epidemic. So previous studies in our lab have shown that a high-fat soybean oil diet can lead to obesity and diabetes in male mice regardless of their age. And this is seen in this graph shown here, where the mice that were on the soybean oil, which is the line in the orange, gain a significant amount of weight compared to mice that were on a standard vivarium chow diet, which is shown in the gray line. And this just kind of indicated that there was development of diabetes and obesity in these mice. And another study in our lab that was examining the effects of both soybean oil and fructose on the development of obesity and its associated comorbidities. And this was in the context of a diet that was moderately high in total fat. And these diets were made from a coconut oil high-fat diet and then coconut oil high-fat diet or soybean oil high-fat diet and then supplemented with fructose. And the total amount of fat in these diets is comparable to that of the average American diet. And in the mice on the high-fat soybean oil diet showed increased weight gain, adiposity, fatty liver with balloon injury, diabetes, and this was compared to the mice on just the fructose high-fat diet. And so if we look at the livers on the mice on the soybean oil high-fat diet, which is the picture on the bottom left, we see these really big red lipid droplets. And this was consistently accompanied by severe hepatocyte ballooning, and this suggested potential liver damage. And so the mice are on the fructose diet. These are the two images that are kind of in the middle. These also showed fatty liver, but not the same type of fatty liver, as they don't have these really big droplets. However, and they're just kind of red more consistently throughout the liver. And so some background on the thiamine project that we're working on right now is to investigate the impact of soybean oil on thiamine uptake and utilization in mammals. And some questions that we asked along our way were, does a soybean oil-rich diet similar to the American diet affect the thiamine pathways? And can using a thiamine analog ameliorate the fatty liver caused by a soybean oil high-fat diet? And so here we have our 4 diets that we used in our experiment. So the first diet is OSD, which is a low-fat diet. The second diet is the low-fat diet with additional TTFD. And if we look on the right, TTFD is a vitamin B1 analog that is used to treat vitamin B1 deficiencies. Our third diet was a regular soybean oil high-fat diet. And our fourth diet was a soybean oil plus TTFD diet to also treat the vitamin B1 deficiency. So here we have doing some body weights and then the liver weights. We have found out that TTFD reduces adiposity in the soybean oil diet in about 16 weeks. So on the graph on the left, we see that the mice that are on the soybean oil diet, which is again in the orange, showed the most weight gain amongst all the mice in all 4 groups. However, the mice that were on a soybean oil and a TTFD diet, they gained a significant amount of weight compared to the 2 low-fat diets, but they did not gain as much weight as the mice on just the soybean oil high-fat diet. If we look at the graph that's on the right-hand side, we see that these are just the liver weights as a ratio of their body weights, and the mice that were on the soybean oil diet had the least heavy livers compared to the mice on the other diets. And this can show that more fat in the liver just leads to it being less dense and it can weigh less. And so for this project, I had to kind of work through this workflow where we start at the mouse and we're able to get the liver tissues on a slide right here. So we're able to stain it. And so to kind of walk everyone through this workflow, we first have to prepare the liver, and we do this by having our groups of mice that are on the different diets that we hope to observe. And then these mice stayed on these diets for about 16 weeks, and then we collected the livers from these mice and placed it in formalin, which is used as a disinfectant and a preservative. And then the tissue was kept in the formalin for at least 24 hours before it was washed in PBS to remove serum and blood components. And then the tissue was kept in a sucrose and PBS solution to further preserve it. And then we were able to embed the tissue, which is essentially to freeze the tissue into these little blocks in OCT, which is the optimal cutting solution, optimal cutting temperature solution. And then when it is frozen into these blocks, we're able to take it to this machine seen right here, which is called the cryostat. And we're able to essentially slice the tissue into really thin slices that are about 5 to 10 microns thick. And then we put them on the slide in this last picture, and then we're able to stain them to look at what's going on these diets. So I focused on 2 types of staining in this project. The first one is Hematoxylin and Eosin staining, which is used to recognize tissue types and morphologic changes. And then I used an oil redo stain to detect lipid content. So here are a bunch of the liver sections that I've stained. Starting on the top left, we have the mouse on the OSD diet, which again is the low-fat diet. We see that all the cells in this liver are pretty closely packed together. There's not a lot of gaps that we could see, which is indicative of a regular healthy liver. This is what we want to see. If we move on to the right-hand side, we see mouse on an OSD plus TTFD diet, which is the additional vitamin B1 analog. Here, the cells on the liver are a bit more spread out. There's more gaps in between them. This can indicate some hepatocyte injury or ballooning. If we move into the soybean oil diet, there is even more ballooning and injury. There are more gaps in between these. We can see like the nucleus more clearly separated from the rest of the cell. We see this even more on the soybean oil plus TTFD diet, where these gaps are the biggest that we've seen out of all 4 diets. And this is very indicative of more hepatocyte ballooning and injury to the tissue. However, we don't know exactly what's causing these kind of gaps, so we did oil redo to test if there was lipid accumulation from the soybean oil diet. So here I have the livers that I've done stained with oil redo. If we look at the top 2 images, these are mice that are on the low-fat diet with TTFD. These images, if we compare them to the 2 images on the bottom, they are not as red. The redness is an indicator of fat in the liver. So we can see that there just isn't a lot of fat in these 2 livers on this low-fat diet. If we look at the 2 soybean oil diets, the diet that was just soybean oil, we can see these kind of like little red droplets in the liver, which shows that there is fat accumulation. However, the soybean oil diet is a little less red than the soybean oil plus TTFD diet. This can indicate an even more fat accumulation and even more fatty liver that is shown in this. So some preliminary conclusions we were able to make were that the hematoxylin and eosin stained sections showed more hepatocyte ballooning in the livers of the mice on the soybean oil diet, and that oil redo staining shows more lipid accumulation in the livers of the mice on the soybean oil diet, and that TTFD appeared to increase the overall hepatic lipid accumulation, although the lipid droplets seemed a bit smaller. The addition of TTFD did not completely ameliorate the fatty liver induced by the soybean oil diet, but it did also improve overall body weight gain. And so I ran into a couple problems within this experiment with sectioning the tissues. There was issues with the OCT separating or being too crumbly. And so we've been working to kind of see if we can re-embed these tissues and if that'll change or if it'll help with the crumbly OCT. And if we're able to do that, then we can move on into confirming these results with more lipid staining. And so for some acknowledgements, I'd like to thank the MARC program and the NIH and everyone in the Sladek lab that has helped me accomplish what I've done in this experiment. Thank you.